Welcome to module 4.2. I am Priyom and I will be walking you through some of a few more CC3D's capabilities. So let me quickly go over the topics that we're planning to cover. So our goal is to construct cells, or more appropriately clusters of cells which have different parts and properties associated with them. And after we have done this, we're also going to look at a plugin called Focal Point Plasticity that allows us to put spring-based links between cell centers to mimic active forces between cells in the tissues. And we'll be doing all of this from the perspective of keeping in mind the biology of epithelial cells. So our goal, one of the goals would be also to construct an epithelium with different, say, apical and basal sites. And we're going to use these FPP properties ultimately to break down these epithelium into different units. So that's what I mean by segmentation there. And the exercises at the end will be focused on whether how well you understand the FPP parameters, and you can also try to calculate the units or the segment cluster sizes, and we'll build up to that. Just as a heads up, so all the codes are available with this link. And you can also download the slides. The slides are on the student material workshop slide deck, and the codes are on in the student material folder under module 4.2 codes. So we're going to begin with the first version 1 code. So either if you want to run it, go ahead and run it. We'll be working with ToEdit to build up version 1 code, and at the end we'll reassess how much time we have to move on. So the code that we'll be working on right now will involve version 1. Okay. So again, just to reaffirm what we'll be talking about, our goal would be to build up an epithelium consisting of different sites, and say an apical and a basal site. And we want to look at properties of construction in which tension on this apical site increases so that the cell adopts a more fold-like morphology. And we would essentially be trying to change the shape of the tissue from, say, a linear chain into a folded one. And ultimately, the goal would be, once we have established the properties that allow the tissue to constrict by using our focal point plasticity plugin, we would also be breaking down an epithelium using these properties of apical constriction. So you guys have already talked about epithelia in the context of lung diseases. And so epithelial cells are basically form barriers protecting us from the environment and lining up vital organ systems. So these epithelial cells, they not only perform a protective role, but they could also aid in the transportation of vital molecules through their bodies. And they could also be involved in, and depending upon where they are, basically, they could be involved in absorption of toxic compounds from the bloodstream. So they have a wide variety of roles. The structures that we'll be building today, learning to build in this module at least, will be of the simple nature, where we just have a single array of epithelial cells. And these epithelial cells are also connected to each other through different types of adhesion junctions, which are extensions of their cytoskeleton network, so that they could extend forces on each other, changing the shape of the tissue. And such processes become very important when you're talking about, say, neural development, where the tissue, where the neural tube folds, and even in the ventral furrow formation, if you're looking at the viewpoint of fruit fly development. So from CC3D's perspective, our goal would be to construct cells, which are epithelial cells, basically, where different regions have different properties. For example, the apical region could have cilia-based processes so that they could sense the external environment, while the basal side would have things like integrins to connect to the basal membrane and connected to the connective tissue underneath. So these different regions in an epithelia, they have basically different specialized functions. So our goal is, how do we represent cells in CC3D that have different properties along a cell body? And once we have done that, we're going to use FPP to basically put links, put forces between the apical side. We're going to pull on these cells and see if we can get those folds that we just talked about. Okay, is the idea clear on what we're trying to do here? Okay. Again, just as a reminder, the slides are up on the slides and the codes are all up. We'll be beginning with version 1. So the goal, what our CompuCell output is going to look like. Let me just show you a quick simulation to that. So, no. Get rid of this one. Let me just stop sharing for a minute so that I can get rid of the poll from my screen. Otherwise, I can't move on.
Priyom, the poll doesn't show up on our screen, so you can just move it aside.
Oh, awesome. Thank you.
Just for your information.
Got it. Thank you. All right. So, So the CompuCell output that we're interested in would look like that to your right. So our cell is going to be comprised of these clusters, where this orangish cell that you see plus this green cell plus this red cell that you see are basically one cell, and it's comprised of these different parts. And our goal would be to create a whole array of them. And these white things that you see on top are what is going to be our FPP links. Okay, so here I'm going to slow down a bit and I'm going to walk you through all the CC3D steps necessary for us to create something like this. The white material that you see on the bottom is what we're going to represent as our extracellular matrix, or ECM in this case, to form a base for our epithelium. Okay. All right. So for this, let's go over to edit. I have listed all these steps in the slide deck, which are over here. So for the first step, what we're going to do is create up a new file. So I'm going to CC3D project. You see, you guys already know this by now. So I'm going to create up a code for this module. Make sure to select Python and XML both for this version. And I'm hitting next. Okay. So if you saw the CompuCell simulation that I just showed you, so you saw that the x-axis had a lot more area than the y. So to do that, I'm going to increase the lattice dimension in the x-axis. So I'm setting that to 300. And along the y direction, I'm setting it to 100. We'll be dealing with a square lattice here, so let's keep it as it is. The temperature, let's keep it at 10. And the index copy range, let's set it to 2. Okay. And we'll be creating our own configuration for this, but there's no option to get rid of these 3 options. So let's just keep it at rectangular slab for now, but we'll be removing this piece of code so that we could create our own configuration. All right. So that's our step 1. So we changed the dimension parameters. We changed the pixel copy range to 2. And that's all we did here. All right. So I'm hitting next. This is where I'm going to add up all my cells. So I want to represent my epithelial cells comprising of 3 different types. So the apical and the basal we already talked about. The rest of the cell body, we're going to create a 3rd type, and we're going to call that as lateral. So let me write down all the types here. So I'm saying lateral, then hitting add to add the cell type. Then I'm adding the apical cell type. And then I'm also adding the basal cell type. So we're going to put all of these cells together to one giant cluster. To represent that extra material that I showed you, I'm also going to create a cell type called ECM. So here I have added 4 different cell types to my list. And I'm hitting next. So the next part, we are not going to add any chemical fields, so we can just leave this blank. Okay, plugins. So what are the things we need? We're going to use contact for sure, where we can establish adhesion properties between different types of cells. Because we're dealing with compartmentalized cells, we're also going to hit contact internal, which allows you to specify adhesion-based properties within a cell cluster itself. We're going to also add links. So we're going to use this new plugin called Focal Point Plasticity. So make sure to click on that. We're going to add volume local clicks, which allows us to demarcate volume-based properties in the steppable file as we write our code. So we're going to add volume local clicks. And along with center of mass on this rightmost column, make sure to click on cell neighbors because we will be counting cell neighbors at different points in the code. So we want to add all of that. So I have 5 extra plugins along with the center of mass that I've added now. So now this is where we're done with our plugin specification. So to edit should generate the automatic XML and the Python scripts for us. So let's hit up finish. And this is going to generate our 3 file outputs. And again, if you feel like I'm going slow, please feel free to download the module code so that you can go at your own pace. So from the to-edit output, what we have an XML file that is going to consist of all the pods parameters and all the plugins that we just generated. We will have a main Python file which is basically going to call the steppable unit that we're going to write down next. And the steppable file is where we're going to manipulate any cell-based properties and into our calculations. So there are a few more things which we're going to change in the plugin file before we can move on to the demo. So let's just go to our XML file. And I'm also showing on these slides to show you what I'm doing. So the first part is the pod section where you have the dimension parameters that we just set along with the steps regarding how many Monte Carlo steps you want to take, the temperature and the neighbor order and so on. And the cell types are all these types that we just created. So this is all fine. We're not going to touch anything here. This is just calling our neighbor tracker, the center of mass, volume, all the extra plugins. What we're going to do next, if people are okay so far, we're going to go to our contact energies. So everything so far is fine. We're going to go to our contact energies, and we're going to change a few of our values. So contact energies are automatically generated so that each of these cell pairs have a value of 10 between them. But considering that this is an epithelium, we want to follow a hierarchy, and our hierarchy is the following. So you saw that Ecm material. We want to say that the basal cell type would like the Ecm most than the other cell types. And each of these cell types like if they're of the same kind, they like each other as well. However, if, for example, if it's a lateral compartment with a basal compartment, they don't prefer being next to each other that much. So that's the hierarchy. So basal likes Ecm, basal will like basal, as lateral will like lateral, apical will like apical. And we can also say that since the apical side is pointing away from everything else, we will also reduce the contact energy between the apical and the medium. So let's make those changes in the code. So if you go to, in this case, the line number that I see, is medium to lateral, so that's fine. I'm setting medium to apical to be 2. Medium to basal is 10, medium to Ecm is fine. Lateral to lateral, let's change it to 5. Lateral to apical is fine. Lateral to basal is 10. Lateral to Ecm is also fine. The other change that we're going to make is the lateral to apical to apical, which we're going to set at 5. Apical to basal, we're keeping it at 10. Apical to Ecm, we're keeping it at 10. Basal to basal, we're changing it to 5. Basal to Ecm, we're changing it to 2. And one more change. In our simulation, we're going to just create one Ecm cell, so that doesn't really matter. But we're going to just set the contact energy between any Ecm cell types to be 0, and our medium cell types, which are also not going to model, to be 0. So those are the changes that we're making. The other thing which if you guys want to explore, would be the next plugin, which is that of the contact internal. So the contact internal plugin, like I was telling you before, lets you model changes, contact energies between cell clusters themselves. So if you want to put any differences between your cell types, you can play with this plugin by changing the parameter values. For the purposes of this demo, I'm going to set all of them to 0 so that I'm allowing my compartments to mix and move freely within the cell cluster. Okay. So I'm setting all of that to be 0. And what I'm going to do next, before moving on to the focal point plasticity tags, is going to go down to the uniform initializer section. So the uniform initializer basically automatically creates the blob configuration for us. So we're going to get rid of the entire tag by doing so. Okay. All right. So these are plugins that you have already seen. I'm going to now go over to the next one, which is something that's new to you guys, which is the focal point plasticity. Now, this focal point plasticity, the moment that you create up the XML file and you call up the plugin, this automatically generates all possible tags between cell pairs, which means that it generates tags between all cell types, and that makes it easier for you to put links between any two compartments. In our demo, we are interested in only links between the apical compartments. So we are going to get rid of all of these tags except for the ones that say apical to apical. The other thing which you have to be careful about is if we want to manipulate focal point plasticity properties in the steppable file, you have to uncomment this tag called local, so that XML knows that the properties of FPP are going to be changed elsewhere. All right. So the plan is we're going to uncomment this local line, and we're going to get rid of all of the other tags with the with different cell types, except for the tags that say apical to apical. Getting rid of these guys here. So the only thing that I'm keeping is this apical to apical tag. So I'm going to get rid of these tags as well, but let me point out, so these internal parameters are again FPP links that you can put within a cell cluster or compartmentalized cells as well. So for example, if you have, say, the lateral, basal, and apical surface within a whole cell, you could also use the internal parameters tag, and they will put the links between, say, a basal and a lateral compartment within the same cell cluster. So parameter tags looks for external connections, and internal parameters looks at links between internal compartments. So since we're not forming any links between our cell clusters, I'm going to get rid of these tags, but you're free to play with these and see if there's anything that you would want to add to the demo. So this is what the focal point plasticity is going to look like. So I have the after the plugin, all I'm doing is I've uncommented local, and I have the parameters saying the apical to apical tag. The XML generates these five different parameters for this plugin. So the first one is the lambda, which you guys have already encountered so far. So that's basically the strength of the FPP links, or how strong the force is between these two different cell types. That's lambda. The activation energy is basically an energy term to make the links more probable, or basically make the creation of the links more probable. And this is only an XML based tag, so we have to set its value here. Okay, so I'm setting its value to -50. Target distance refers to the distance that you want the two center of masses to take, or basically your desired distance between the two center of masses that you want to achieve. Maximum distance is related again to the link. Basically, it tells you over what distance is permissible for the two cells to form a link. And the max number of neighbors basically tells CompuCell on how many number of neighbors or how many links each cell is going to form. So since we're going to manipulate all of these properties in the steppable file, I'm going to set all of these parameters to 0, except for the activation energy, which is an XML-based tag. So lambda is set to 0, target distance is set to 0, max distance is set to 0, and I'm also setting the maximum number of junctions to 0. Okay, and that's all we have to do for the XML. I hope everybody got that. If not, please look up the slides where all these steps have been listed. And don't forget to save it. So we're now done with our main Python XML file. The main Python file, we're not going to be making any changes here. So we're going to now move on to our steppable file. Okay. Right. So what are the things that we're going to do on the steppable file? So let's begin by first creating a base for our epithelium. So what we're going to do is start by creating up this Ecm material that I showed you. Okay, let me just check on. All right. So what I'm going to do here is I'm removing these comments under the start function. So I'm now in my start function. And my goal is to create up this Ecm-like material here. And to do this, I'm going to go to CC3D Python. I'm going to go to cell manipulation, and I'm going to go to create cell. And so this generates a snippet for me that assigns a new cell type, a new underscore cell, to the cell field properties that allows me to define what regions of the lattice I want assigned to this particular cell. In this case, what I want to do is I want to create an Ecm type that I have described in the XML. So under type name, I'm going to put self.ecm, and the dimensions of this are going to be dictated how big or how small I want the cell. I can set it using the cell field function. So in this case, my lattice size in the x direction, that's about some 300 pixels. So I'm going to do 0 to self_dim.x. So this self_dim.x command lets me find out the maximum extent of the lattice in the x direction. I could also very well set it to 0 to 300, and you would basically get the same result. So this is 0 to self_dim.x. And in the y direction, I'm going to cover about half the pixel dimension. So I'm going to cover half the pixel size, half the lattice size. So I'm going to go from 0 to 50. Okay. Another useful thing is sometimes if you want to copy the cell to a variable, then it might make it easier later on to assign certain outer properties to the cell type. And we'll be doing that when we define our lateral, basal, or apical cells. So let me just copy this Ecm cell, new cell to a variable, and we'll just call it cell. And I'm just going to copy this tag to this so that I refine the name as cell. Okay. All right. So at this point, if you open up CC3D and you step through, and you just step through one Monte Carlo step, this is the configuration that you're going to see. In the interest of time, I'm not going to stop at every instant and show the output to you, but these are the checkpoints that you can take to make sure that you're getting the same sort of simulation. Okay, so far what we have done is define the Ecm cell. Okay. All right, now we're going to define 3 different cell types for our epithelial cell. I've already told you that we're going to define a basal, lateral, and apical site. So let's create those cells. And I'm going to be copying that new cell tag that I just showed you. So I'm going to say lateral is a new cell. And the type of this new cell is lateral. Then I'm going to create a basal cell. And I'm going to assign it to the type basal. And I'm going to similarly do it for my apical site. Okay. And here I'm only creating them, so I need to assign them to the lattice. So I'm going to again do this by using cell field. So let's first assign the lateral cell. By the way, I'm trying to create cells which are of a height of 20 pixels and a width of 10. So that's why you will see that and the width is along the X direction. So that's why you might see slightly taller cells in the Y dimension. So I'm doing self.cell field 0 to 10, which is along the X direction. And I want to place the cell on top of that Ecm material that I just created. So since the Ecm ended at the 49th pixel, I'm going to start from 50. And I am going to assign it a slightly smaller volume. So let's put 54. So doing this, so 50 to 54 and 0 to 10, gives me a cell volume of 40 pixels. So each of the basal and apical sites that I'm creating will have 40 pixels, and the lateral cells that I'm creating will have about 120 pixels. Okay. So this is being assigned to basal. And note that I'm going from the bottom up, so I'm following where the Ecm ended. So the next thing I'm going to do is now assign the lateral volumes, which is again starting from the same position. Now I'm going to do 54. So I want about like 12 pixels in this direction. So I'm doing 66, 0. That gives me lateral. And finally, I'm doing apical on top of these two. 6 to 70. Okay. So now at this point, if you step through one Monte Carlo simulation of your CC3D code, this is what you should get. So we have the Ecm, and we have the basal, lateral, and apical cells on top of it. However, if you notice at this point, there is no talk of compartmental cells yet. We have like created these 3 cells as different cell types, so they don't know that they belong to the same cluster. So our goal next is going to be to assign this basal, apical, and lateral cell to the same cluster of cells. So we're going to do that by the following. So if you go to CC3D Python, let me just create a bit of a space here. Okay. If you go to CC3D Python, go to cell attributes, and then you have this code called cluster Id reassignment. So if you click on it, this should give you the code to reassign a cell to a chosen cluster. I'm going to just get rid of these comments because I want to show you all the pieces of the code at the same time. So I'm removing the comment, and this is the line I'm interested in. So this is the reassign cluster ID, and here I'm going to reassign the basal cell to a cluster ID. Now this cluster ID could be any of these, related to any of these cells that you want to choose. In this case, I can choose this cluster ID to be the lateral cell. So this cluster ID is a new object. So you guys have probably seen cell.id so far. So this cluster ID, because the IDs are intrinsic to the cell type, you can additionally define a tag called cluster ID, and you can reassign these cells to this bigger cluster ID tag. So that's what I'm doing here, that I'm reassigning the basal and apical to the lateral cells cluster ID. And you could even do any of these combinations. For example, you can assign lateral and apical to the basal cells cluster ID, or the basal and lateral to the apical cells cluster ID. So you can pick up any of these combinations. As long as all three of them are part of the same cluster ID, they will behave as a unit. So that's what I'm going to do next, where I'm also going to reassign the same for the apical cells. Okay. So now what we have done is these cells are now created as part of a cluster, and I have associated them to the lattice. The next goal is basically to complete the epithelium. So here I have just created one cell at this point, and you can run this again, step through one simulation time step. You should be able to see one cell on top of the ECM. My goal would be to create a whole array of epithelial cells. And to do so, I need some sort of a loop so that it can iterate over my cell objects. So what I'm going to do now is, I have this basic code here, which creates up these cells. I'm just going to loop them over with a for loop. So I'm going to say, again, so my width in the X direction was about 10. I have about 300 pixels in the X direction. So I can put about 30 cells. So that's what I'm doing here. I'm saying that for num cells in range 30, create all of these cells. Okay. And I guess many of you can see the problem right away, because I'm not changing any of these cell field parameters. So every time, even if I loop through this, all these cells are going to be created at the same position of 0 to 10 and 50 to 54 and so on. So I need something that basically iterates through these cell field positions so that I could get these cells at different positions. So I'm going to create a placeholder, a variable. I'm going to call it X position. And I'm going to begin that with 0. And I'm going to iterate through my cell field arguments with this X position. And so I'm starting with X position. And every time X position varies, I change it by 10 pixels. So this again defines the width of the cell. And I'm going to do it for all of these guys. Okay. And make sure I have to keep changing this X position in the for loop. So I'm also going to increment this with this 10 pixels. And you can do it your own way if there's any other way that you find it comfortable. Our goal is to just create up an array of cells along the x direction. Okay. So if you again step through one Monte Carlo step now, you should see that an array of cells have covered up the x dimension on top of the ECM. However, we're missing one important piece, and that is the volume. To define the volume properties, what we're going to do is use our lambda volumes. So let's define the lateral cells properties first. So let's say that the lambda, lateral dot target volume. If you can see from the cell field arguments, the lateral cell has about 12 pixels in X and 10 pixels in Y. So that's, 12 pixels along Y and 10 pixels along X. So that's about a 120 cell volume in total. And then I'm going to also add the target constraint for this, which is about, let's keep it at about 20. And then we're going to do the same thing for the other two cell types, which is basal, target volume. And here we assign some about 40 pixels. Okay. Right. So that takes care of most of our issues. In reality, we want the epithelium to be bounded by something, because in real life, you will have tissues or structures that limit the size of this epithelium. So what we could do is implement some sort of a fixed boundary on either side of the epithelium. And the simplest way we could do it is just say that the cell at the beginning and cell at the end are mostly like frozen. So they behave like some sort of a wall on either side. And while this is not going to be that important for this exercise that we're doing when we go over to segmentation, it will slightly help us to have these fixed boundaries on either side of the epithelium. So what I'm trying to do is, I'm saying that if the epithelial cell that I designed is either at the first position, shown by an index of 0, or it's either at the end, then make the lambda volume of these cells a much larger value so that they remain mostly stiff and immobile, not making any pixel copy attempts. So here I'm increasing it to 100, and that should basically keep them fixed. Okay, so now you're free to, if you have written through this code, you're free to run it, and you should have the first and the last cells frozen mostly, and the rest of these cells, because we have now defined their volume properties, they should be preserved, and the simulation should be working. Okay. Next, what we're going to do is add focal point plasticity parameters to our simulation. And what we're going to do is define links only between the apical sites. Okay. So let's do that. I'm going to, if people are okay with this code with the for loop, I'm just going to minimize the snippet so that I could add the focal point plasticity in the same page. Okay. Oh, I am missing an equal to sign here. Yeah. Don't forget to, the if conditions use double equal sign, so don't forget that. Okay. So I'm minimizing this. So now what I'm going to do is I want to add links to all my apical sites. So I'm going to visit all my apical cell types. To do this, I could use my two edit tabs. I could go to CC3D Python. I'm going to go to visit, and I'm going to go to all cells of given type. And this is going to give me this piece of code that allows me to visit a particular cell type. And this is slightly indented, so let me get this back so we can see. Right. In this case, the cell type that I want to visit is apical, because that's where I want all my links to form. So I'm going to replace this type name by just apical, and I'm going to get rid of all these under text. And now, once I have found the list of all my apical cells, what I'm going to do next is add the focal point plasticity links. To do this, I need to find out my nearest neighbor for each of these apical cells, because I'm forming links only between neighbors. So go back to CC3D Python, go back to visit, and then click on cell neighbors. This should give us the snippet for finding neighbors between any cell types. In this case, we are only interested in apical neighbors. So what we're going to do is we have visited all the apical cells. We have found out all their cell neighbors. If we have established that it's a cell type that's a neighbor and not the medium cells, as denoted by the if neighbor tag, we're going to go inside this loop now. I'm going to get rid of all of this other text because I don't need it. And under if neighbor, I'm going to say that if you are another apical cell, then we'll form a link. And to form that particular focal point plasticity link, go back to CC3D Python again, go to focal point plasticity, and go to create FPP link. Okay. Once you've done this, this will give you a tag with self focal point focal plasticity plugin create focal point plasticity link, and with all of these parameters that we saw in the XML tag. So cell 1 and cell 2 are the cell and the neighbor that you want to form the link between. So I'm going to replace this by the cell. Cell 2 is the neighbor, so I'm going to say neighbor. And lambda and the target distance based properties are, we're going to just define them in a moment. The maximum distance, in this case, let's set it to equal to the dimension of our lattice as 300. So in this case, let's pick up two values of lambda and the target distance that we want to achieve. So let's set lambda fp to 10, and let's set the target distance between these to 6. All right. And once you have done that, we're going to input this in our so this is lambda fp. And I'm replacing this by target distance. So far, all I've done is I've called up the focal point plasticity plugin. I have found out all the apical cell types and their neighbors. And I have used this focal point plasticity plugin to put a link between these two guys, effectively a spring, which has the target distance of about 6 pixels and the lambda constraint at about 10, lambda fp of about 10. So if you run the simulation now, what you should see, and I know it's probably hard from these slides to see that, you should see a white line between all of the apical cell compartments, indicating that the link has been successfully created. Okay, so in the start section, we have created all of these FPP points. Now we're going to next go in and try to manipulate this FPP plugin so that we could get the tissue to fold. Okay, so that's our next goal here. I hope everybody is with me so far. If there are any questions. All right. So
someone asked to repeat the very last step.
All right. So what we're going to do is going to create up the focal point plasticity links between our different cell types. So in our case, we only want to increase tension or define tension-based properties on the apical side. So we're going to create up links only between apical cell compartments. To do so, what we're going to do is find our list of all our apical cells, which we have done here by saying cell list by type, self-apical. This is going to create these cells belonging only to the apical cell, apical sublist. Then we're going to find out all the neighbors that are present for each of these cells. If you have a neighboring cell that's also of the apical subtype category, you're going to form a link between them. At this point, the link properties that we're forming will have all uniform properties. So all these cell pairs that we have created will have a uniform lambda FP and a target and a uniform target distance. I think I have the CC3D code open that I could show it to you. So this is what I mean. So these links that you see are between these different apical cell compartments, and that's being created by using this create focal point plasticity tag. Okay. Once you have this, then we're going to now move on to the step function. So if you run the simulation, so nothing should really change because you have equivalent lambda properties and the equivalent target distances between all of these cells. And so you will only see the additional presence of these links now. We're going to next go and see if we can manipulate these FPP properties so that we can get the tissue to fold. So to do this, let's go to our step function. And instead of creating these folds right at the beginning, let's give our simulation some time to settle down. And say about around 500 Monte Carlo steps, we're going to impose this asymmetry properties on these cells to get them to fold. So we're going to say if the Monte Carlo time step is 500, let's go to our cell list again. Let's visit our apical cell types again. Once we have collected all our apical cells, let's check if they're within a particular distance. So we're not going to constrict everything at this point. Let's just say that we're going to impose certain differences in our FPV parameters only between a certain distance. So let's say if the cell lies between, say, 200 pixels and 100 pixels, then we're going to find out the links basically that this cell has. So we're going to go to CC3D Python. We're going to go to focal point plasticity, and we're going to go to properties. And if you hit on properties, it's going to give you the focal point plasticity links and tags that's associated with that cell type. So what we're trying to do here is manipulate these FPP properties within those particular X pixels between 100 and 200 to have different properties than the rest of these cells. So if your cell type is within this, then get the focal point plasticity properties associated with that cell type, which we're doing here by adding this tag for FPPT in self get focal point plasticity data list. That's the tag. So basically this list contains all the FPP properties that's associated to this cell type. I'm going to get rid of these information here. And I'm going to also get rid of this. So what I want to do is in that tag, for setting the focal point plasticity, I want to change some of these parameters. So the create focal point plasticity link creates the link for the first time, and the set focal point plasticity parameters allows you to modify or adapt the focal point plasticity parameters. And if we have time, we'll hopefully also see how to delete these focal point plasticity links as well. So I'm going to set focal point plasticity parameters. So this is between the cell, the apical cell that I picked up, the focal point plasticity data list for each of these cells contains a tag called .neighbor address, which is basically the ID of the neighboring cell to which it's linked with. And these other two tags are again similar to the lambda FP, basically the strength of that constraint, and then the target distance. And the third tab is your maximum range over which these links are permissible. So we're going to set that to 300. And this is where I mean inducing differences in the FPP properties. So I'm going to say that if you're a cell and you have your neighbor within this distance, I'm going to increase that strength of that constraint to 70. And I'm going to increase the target distance. Sorry, I'm going to decrease the target distance to about 4. And I'm going to make sure that the focal point plasticity takes my new parameters. So I'm going to write lambda FP here. And I'm going to put target distance there, right? Lambda FP goes under the strength of the constraint and then the target distance. Sorry, I don't know, you can't probably see my whole screen. Maybe I can try to make it smaller. Okay. All right. So what we've done so far is in these initial step, we had found out all our apical cells, found out their neighbors, created this focal point plasticity link. And then in our consequent time step, we try to manipulate some of these focal point plasticity parameters using set focal point plasticity. And in between those couple of cells, what we did was we increased the target, we increased the strength of the constraint, and we reduced the target distance between those two cell types. And now what we're going to do is we're going to run this demo. So feel free to run the version 1 that's uploaded online, or you can run up the code that you wrote so far with me. And if you run the code, so initially nothing happens. So these cells are subjected to fluctuations with this temperature parameter. So nothing interesting happening, but around 500 Monte Carlo steps, what you see is that the cells are pulling on each other because they have an increased constraint strength between them. And they're also trying to minimize the distance to about 4 pixels or so. And when they do it, there's an increased tension on the surface so that these cells have adopted a more constricted shape. Or in other words, you can start seeing these folds appear in the tissue. Okay. So one of the goals of the exercises was to let you explore and play with the FPP parameters. So you could try increasing the lambda FP in this region to say to say 80 or 90, and also try reducing it to say 50 or 40 and see what differences in the profile that you get. And you could also try changing the target distance to a smaller value, say to about 3 pixels or so, and see what kind of profiles you get. So I'm going to give you a minute to play with the simulation while I assess how much time we have and how we want to proceed with the next segmentation demo.
Priyom, how much more do you have for this?
So the next plan was to show segmentation using the properties of apical constriction. So here where we have activated constriction in cells simultaneously, the goal was to implement some sort of a wave to constrict them in pairs and then break them after reaching a certain threshold and to see segmentation. And one of the goals would be basically the exercises at the end was to calculate the size of these clusters as an exercise. So it's up to you. So one thing we could do is either not walk through the code, but I could just explain the code that we have and let people work on the exercise. Or, I mean, it's up to you how you guys want to proceed.
I think we want to give Gilberto at least a little time to show his demo. So maybe one more, and then we'll leave the rest as an exercise. So if you can put that into the exercises docs file, that would be great. There was a request to show the steppable file, and there was a comment that somebody was having a crash with the player.
So the code has been written using version 4.2, so it should be compatible with NanoHub, which I've checked that it works. Maybe Giuliano or somebody can check that out while I move on to the next part. Okay, so what I'm going to talk about now is quickly just give you an idea of, so we build the epithelium, and we know how to use links to basically constrict or change properties at the level of these cells. We can also use these links to basically segment or break a tissue. And I'm just going to walk you very quickly through the code. And what the goal for you tonight would be to determine the size of the clusters that it form. Okay, so let's just do that. So to do this, we're going to focus on two things. Instead of implementing simultaneous constriction like we did for a bunch of cells between 100 and 200 pixels, one of our goals would be to activate cells sequentially. And so we want to build up some sort of a wave of constriction that starts off with, say, the left end, and then every couple of Monte Carlo steps progresses to the next cell or so and tells it that it's time to start constricting. So that was the first goal. And then we're going to say that when this is happening, you have seen that the tension profiles or how the tissue folds depends on the parameters of lambda, that strength of that constraint, and the target distance. So we're going to use a criteria based on the combination of those two, which we're going to label as tension. And we're going to say that if the tension in the tissue exceeds a certain threshold, then the tissue is, then the cell links are going to break. And wherever those breaks occur, you're going to get a cluster or a group of cells. So let me basically then zoom in on my slides. So that's the basic idea. And the goal was to introduce two sorts of dictionary attributes that allows us to model this wave. And we would have called them constrict and graph. And as we move over to the exercises, one of the goals would be to use this dictionary attribute so that you can define the cell clusters on your own. So this graph is simply there to let you build towards measurement of these clusters. And so the idea is that let's label all of our cells with this constrict tag. And what we're going to do is, to understand how to model up this wave, we're going to create up these two lists. One is a list containing all of our cell IDs of our apical cells, and the other would be a list where we would keep store of the links that would eventually break. So that's the step where we're just storing all the apical cell IDs so that we can model our wave, or the sequential pairwise activation process. And we're doing this in this step function here. If you want to run this part, you can go up to module 4.2 version 2, which basically runs the sequential activation code. And so the idea is that every time you reach a certain number of Monte Carlo steps, in this case, I have chosen it to be at 300, a new cell is activated, and I'm choosing these cell IDs from that list that I just created, which is the self list IDs. And I'm going to activate constriction by saying that constriction becomes 1 every time that Monte Carlo step time has been reached. Okay, and then I'm going to keep doing this for all my cell IDs in that list until I reach the end of my apical cell list. Okay, so this is what this part is doing. The other thing is, usually to implement these codes, it's a good idea to wait for a while. So that's why I have this relax time tag added, so that I only start applying my wave after waiting a certain period of time. So I've waited like 50 Monte Carlo steps, and then I have started sequentially trying to constrict my cells. So here I'm only keeping track of all my cell IDs. I have not changed any of your focal point plasticity properties yet. And this is where I'm doing it next. So the idea is, once you have cell and its neighbor that have been passed by that wave of constriction, if both of your cell ID has received the okay to constrict and its neighbor has received an ID to constrict, then we're going to change the focal point plasticity parameters so that I'm going to start increasing this lambda parameter by, say, 0.05. And you can change the parameter and also see what other kind of profiles you get. And we're going to keep the same target distance, because it might be difficult to understand the profiles by changing both of them at the same time. So we're keeping the target distance fixed at the 6 pixels, and we're just changing the lambda, the strength of the constraint by 0.05 in every in every time step, and we're setting all of our focal point plasticity parameters as this new lambda and as a new target distance. Now note that the only other thing that I've added here is that I'm only checking once, which means that I don't want to pick up, say, if I have cells labeled 1 and 2, and I'm putting out 3D loop, I only want to check once for 1 and then 2. And I do not want to again sweep when it comes to 2. I do not want to again go back to 1 to increase up this lambda parameter. So that's why I'm just making sure that this loop is entered once by checking up these cell IDs. And again, you might have your own way of implementing it, and feel free to do so. So all I've done here is found out my apical cells, which have been passed by that wave of constriction. And if that cell is ready to constrict and its neighbor is ready to constrict, I have increased the target length. I have increased the target strength of that FPP constraint. And I'm keeping the target distance in here. And so you should start seeing profiles where these cells should start grouping out as this lambda is increased. The next thing that we're going to implement is we're going to say that because these cells are grouping together, these cells must also be under heavy strain and tension. So when that happens, we're going to say that these cells can endure these links only up to a maximum criteria. And we're going to calculate a criteria based on our lambda times the target distance between them. And one of your assignments would be to change this criteria and see what kind of other profiles that you get. Here, what I'm calling this criteria as a tension-based quantity, where I calculate this lambda times the distance that these cells are able to maintain minus the target distance that was intended for them to maintain. And this quantity gives me a metric of how stressed these cells are. And so I'm saying that if these cells exceed a certain threshold of the stress, then start breaking the links between these different compartment cells. Okay, I know this is a bit quick, but I hope if you look up the links, PDF, it might be a little bit more clearer. So that's the criteria that we're going to adapt. Where here we have put the number specifically at 400, but again, this is something that you can vary in your simulation and see what kind of clusters that you get. And once I have established the criteria, I'm going to go over and use a focal point plasticity property called delete focal point plasticity link, where I'm going to break the links between cells if they exceed that threshold. So that's all I'm doing here. And deleting the focal point plasticity link is going to remove that those links from my CompuCell demo. And once you follow these steps, or if you run this version 2 of the demo, what you should see are segments like this, where here are the position where the links have been broken. And so the epithelium is now segmented and has been broken down into these different clusters. And so all we did was we checked out how much tension existed between the cell and its corresponding neighbor. And we picked up a criteria that said that if the tension exceeded threshold, then start breaking these links using focal point plasticity parameters. And we ended up with these clusters. So one of the exercises was, if you have version 2, please use it to change the tension threshold to 300 and 500 and see what differences you get. And you can also fix the tension threshold and see if you can measure the sizes of these clusters. There is a code called version 3 in the code module, which you can use to check out your solution. And you can use the dictionary attribute graph in this case to basically help you find out where this link breaks have occurred. Right. I can talk on chat for any other questions, and I'm going to just stop here and turn this over to James.